Hi everyone, my name is Harjap and today I'll be talking about the convergent evolution of eyes across the tree of animals. So this is how I chose to break down my presentation. The first part will involve me talking about the basic ingredient necessary for eyes to form in animals later on. Next, I'll talk about the evolutionary history of eyes and their convergence. And the final section of my presentation will entail me examining different types of eyes that we see today. Finally, I'll wrap everything up with a nice conclusion and share a little figure that showcases a potential pathway for how eye evolution took place over time. Okay, so the formation of the eye involves several different components, such as needing a cellular mechanism that allows for the absorption of photons, a process by which photoreception can be processed by an organism, and a genetic blueprint for creating such a sensory structure. Light is a physical property, and organisms first needed a way to capture light in order to then be able to utilize it for a specific function. Organisms like cyanobacteria, as seen here, were able to develop chloroplasts through an endosymbiotic event, and this allowed them to capture sunlight to produce food, as seen in autotrophic species today. This process eventually allowed for beta carotene, a derivative of vitamin A, to be produced, and this compound is essential to the production of retinal. Retinal, as shown in the bottom right of your screen, then became the chromophore present in all types of eyes that is able to absorb visible wavelengths of light. Another eye component which is homologous across all types of eyes is the protein called opsin. Opsins are G protein coupled receptors, also known as GPCRs, and when these transmembrane proteins are paired with a chromophore like retinal, which can absorb light, opsins are then able to produce an intracellular biochemical signal that allows for light to be processed. Although in the beginning, opsin-like compounds covalently bonded with retinal in a cell with one or two cilia and acted as a proton pump for such early archaean species, over time, these retinal opsin compounds, called rhodopsins, congregated together into an eye spot where multiple light-sensing containing cells of rhodopsins allowed for these organs to detect whether or not they were present in light or in darkness. In addition to rhodopsin, a so-called master gene has been found to be involved in the eye construction of several species, from arthropods like Drosophila to vertebrates like us. The PAX6 gene acts as a master regulator in eye development across diverse metazoan species, and the predecessors of the PAX gene family, like PAX-B, can also be found in periferins and cnidarians that are able to detect light. This gene, along with others in the retinal determination network, not only initiate eye formation, but they show deep genetic homology across animal groups. Homologous genes like PAX6 not only originated in early metazoans, but they've been preserved over time, with evolution adapting them and their specific visual needs for different lineages. This genetic conservation across species highlights how a shared ingredient list allowed for different lineages to independently develop complex eyes, each adapted to their own unique environments. In the previous section, we learned that photoreceptive compounds like vitamin A-derived chromophores and their associated opsin proteins are homologous across animals alongside the PAX6 gene, and all this allowed for the formation of eyes. So then why isn't the origin of eyes considered to be homologous across all animals, and why does the scientific community firmly believe that eyes independently evolved at least 40 times throughout evolutionary history? In this section, I'm going to delve into how different types of eyes emerged during the eras of time, and these eras of times have been highlighted by me here in blue rectangles. We'll be exploring the Proterozoic era, the Cambrian period, the Devonian period, the Carboniferous period, and the current period of time. The story of eyes begins in the Proterozoic era, which took place between 2.2 billion and 543 million years ago. The first light-sensitive molecules likely appeared in single-celled organisms that were swimming in the ocean. These simple organisms could detect basic changes in light via their eye spots, which gave them a survival advantage, helping them to move towards sunlight to find energy. The first type of eye-like structure likely appeared within protists that resemble dinoflagellates as seen here, as they appear to possess ocelloids in the lower half of their body. Ocelloids are pigmented cups containing retina-like and lens-like structures. We still don't fully understand how these tiny organisms that are only about 70 micrometers in length were able to process vision, but them lacking a brain and having these eye-like structures suggests that eyes emerged prior to any neural processing center being present. Cnidarians within class Cubozoa that gave rise to modern-day box jellies also emerged sometime during the late Proterozoic era, and although we're not fully sure if these early cnidarians possessed eyes, their descendants most definitely do. Box jellies possess structures called rhophalia, and these typically contain around 6 eyes. A closer examination of these rhophalia reveals that some of these 6 eyes, like camera-style eyes of our own, can be seen here alongside simple pit eyes as well. The Cambrian explosion took place about 540 million years ago, and this marked a period of intense evolutionary pressure where predators and prey were competing intensely for survival. Complex eyes began to form in this era, evolving independently in different lineages still seen today, like arthropods, mollusks, and chordates. The first known animal eyes emerged during the Middle or Early Cambrian, belonged to arthropods known as trilobites. Trilobites, as you can see here, possessed compound eyes that had calcite composition of their ommatidia, which are individual units of their compound eyes, and thus this composition of their eyes didn't require fossilization to be preserved. During the Cambrian era, animal life was vastly abundant, and predation likely played a big role here in driving eye evolution, as animals with better vision had an edge in both hunting and in avoiding predators. Next, we have the Devonian period, also known as the Age of Fishes. This period of time lasts between 450 million years ago to 362 million years ago, and it led to more advanced eyes appearing in aquatic species. Fish developed eyes with lenses and retinas, allowing them to develop greater vision quality with an increased range and spatial acuity with sharper focus. Both of these were crucial features for a predatory lifestyle in complex underwater environments. These fish would develop camera-style eyes best suited for their aquatic lifestyles, as they had fixed pupils, and as you can see here, a large round lens accompanied by lens movement, and a flat cornea with no refractive abilities, as its index of refraction is the same as water. Not only this, but animal species also began to emerge on land during this period of time, which required them to gain their own set of adaptations for their eye structures. So, extant amphibians like frogs serve as really good examples for how these eyes transition from water to land. The cornea became steeper and clearer as the refractive index of the cornea and air is vastly different. Another big change believed to occur was related to stereopsis or depth perception, which is dependent on visual inputs from each eye going to both sides of the brain. Analysis of early amphibian species like frogs suggests that tadpoles had a completely crossed chiasm, which is similar to the completely crossed chiasm you see here in this figure for fish. And this prevented them from having binocularity. But as tadpoles undergo metamorphosis into adults, their chiasm is decussated, leading them to have enhanced visual processing on land. And this can be seen in the various other vertebrate chiasm you can see in this figure here as well. Such an event is likely what occurred for the ancestors of all modern-day tetrapod species as they transitioned their eyes from an aquatic lifestyle to a terrestrial one. The Carboniferous period lasted between 362 million years ago to 299 million years ago. And cotylosaurs, the stem reptiles, branched into sauropsids and synapsids, establishing the two primary lineages for terrestrial vertebrates. Sauropsids, including modern-day reptiles and birds, developed a more efficient form of eye accommodation using lens deformation rather than movement, offering faster and more accurate visual adjustment. This adaptation allowed animals to produce a clearer image, but snakes, a subgroup of sauropsids, reverted to lens movement as they emerged from an underground lifestyle. Similarly, synapsid evolution led to various mammalian lineages with diverse adaptations in visual mechanisms, ultimately influencing the complexity of color vision seen in later mammals and primates. In both sauropsid and synapsid lineages, adaptations in color vision evolved progressively, with early tetrachromatic abilities seen in reptiles and birds, while later in mammals, especially primates, they developed trichromatic vision properties. So even though I've mainly discussed sauropsids and synapsids having color vision, don't be mistaken that color vision is only present in these animal lineages, as many insects are also capable of seeing a wide array of colors. So now moving on to the present day. I want to showcase this figure here that shows the current diversification seen in current day eye morphologies, with a specific focus on molluscan lineages, as molluscan taxa provide a wide variety of different types of eyes relative to other animal phylums. Some other phylums seen here are Platyhelminthes, which have their cup eyes, and the camera eyes seen in most vertebrate species. And as you can see here for the mollusks, you can see mirror eyes, compound eyes, and cephalopods have both camera eyes and pinhole eyes. Some animals that aren't shown here also have distinctive eye morphologies as a result of convergent evolution, including the 6 different types of compound eyes seen in arthropod species alone, which I'm going to mention later on in this presentation. So now, this final section will be highlighting the main types of eyes we see in animals today, showcasing the range of visual strategies that have evolved. This is a great place where I also want to mention that lens proteins utilized by different animal lineages for their eyes aren't homologous. However, the strategy for making these lenses is. And this, in return, really showcases how these eyes adopted convergent solutions for reaching the same conclusion for their eye formations. Despite all of this, in this section, I want you to understand how different types of eyes, like cup eyes, pinhole eyes, compound eyes, and camera eyes, all emerged and are present here today. So we're going to begin with the eye spot. An eye spot is the simplest form of a light-sensing organ, and even though it's not technically an eye, I still want to talk about it because of how prevalent it is in small organisms, including some small myofaunal animal species. It can only distinguish between light and dark, but that's enough for these organisms to orient themselves in their environment. This primitive structure represents the foundation from which more complex eyes evolved. As I mentioned earlier, it's believed that over a period of 35,000 generations, the eye spot of an ancient organism developed a concave cup surrounding the eye spot, leading to more refined vision being possible. The curvature of cup eyes, as seen in flatworms, allowed them to be more refined, and this allowed them to be more fitting of the function of a traditional eye, as it made it possible for spatial information to finally be detected in their collective environments. Moving on, we have the camera eye, which is found in vertebrates like us and in cephalopods like octopuses, and these provide detailed images. These eyes are essentially still simple eyes, like cup eyes, but with added elements such as a cornea, a lens, and extraocular muscles that help animals within their specific ecological niches. This type of eye has a lens that focuses light onto a retina, allowing for sharp, high-resolution vision, and this is the exact type of eye you're currently using to view this presentation. Despite evolving independently, the camera eye in cephalopods and vertebrates is strikingly similar in function and form as seen through this image on the slide. You can see the developmental pattern for 3 convergent eye types in this picture, with vertebrate camera eyes in the top row, the simple eyes of spiders in the middle row, and the cephalopod camera eyes in the bottom row. Notice how both different types of camera eyes are vastly different in the beginning of the development, but as you progress to the right, you can see that they adopt similar structures like a retina, lens, and cornea, while also developing to possess a strikingly similar shape. Next, I want to talk about compound eyes, and these are very common in arthropod species. And basically, compound eyes consist of thousands of tiny lenses called ommatidia, which I mentioned before for the trilobites. And essentially, each ommatidia is able to capture a small, low-resolution image of the visual field. This eye type excels at detecting motion, providing a wide field of view, perfect for fast-moving insects. Compound eyes are themselves classified in 6 different morphological categories, and some of these categories can be seen here in this image. These include apposition compound eyes, afocal apposition, neural superposition, refracting superposition, parabolic superposition, and reflecting superposition types of compound eyes. Each of these compound eye categories differ in their type of image that the animal sees. Finally, I want to talk about some other unique eye types that have evolved to meet their specific ecological niches. And for this, I want to use this figure as a base to showcase 8 general optical patterns that have emerged for single-chamber eyes during the Cambrian for 3 specific phylums, Arthropoda, Mollusca, and Chordata. A indicates the pit eye, which was likely present within the common ancestor of these 3 phyla, and this was likely the beginning point from which the other types of single-chambered eyes developed, as indicated by the large black arrows. On the other hand, the small black arrows showcase the pathway for light that light takes to enter any of these respective eyes. The pit eye likely developed into the pinhole eye of Nautilus with no lenses, as shown by B. The mirror eye, which reflects light onto the retina of scallops, is seen in I. And the common single-chambered eyes with a singular lens seen across many, many species indicated by C. F and G then showcase eyes found in species with multiple lenses like Pontella and Coppilia, which are both arthropod species. Next, D shows an eye with homologous lens, which cannot focus, whilst E shows an eye with a lens with a gradient of refractive index. Finally, H shows the terrestrial eyes possessed by us, with the protrusion showing our cornea and the small circle representing the lens directing the light towards our retina. So, that was a lot of information about eyes, and just want to summarize what we've learned today in this presentation. Firstly, homologous elements are present within almost all animalian eyes. The presence of chromophore-like retinal combined with opsin proteins and the Pax family genes provided animals with the necessary structures which would allow further eyes to form. Secondly, we explored how evolutionary pressures across time led to different animal lineages evolving eyes that were helpful for their specific ecological niche, which is why we see a wide variety of eyes today across metazoan species. Finally, we thoroughly examined the different types of eyes seen today and how simple pit eyes develop into complex eye types like compound eyes and camera eyes. Before finishing this presentation, I just wanted to show this final picture, which I believe really greatly encapsulates the landscape for how eye evolution took place. Essentially, in this image, optic quality gets greater as you go from the bottom, from where there's just mere photoreception, all the way to the top where you have eyes with excellent optical quality, like the 3 different types of compound eyes on the left and the well-developed camera eyes of fish and vertebrates. But what I love the most about this image is that in order to get from one type of eye to the other, you must follow the black line, meaning that you simply can't just jump from, for example, apposition eyes to this eyes possessed by spiders. But yeah, that was something I really found really cool and just want to share with everyone. And thank you again for listening to my presentation.